Hello, Thomas Wimes here from the Santa Barbara Nutrients YouTube channel. I had a chance to interview a very special person who is of great importance for the research field in polycystic kidney disease, Dr. Stephen Reeders. He was in town here in Santa Barbara, and we had a little walk along the waterfront. So I asked him about the history of the discovery of the polycystin-1 gene. That's a gene that's affected in most patients with autosomal dominant polycystic kidney disease. And so Steve, Dr. Reeders, was actually the pioneer many years ago that kind of led the effort to identify this gene. And so in the interview, he explains how this all happened, what the kind of the backstory is, who the other scientists were that were involved in the effort. And Steve Reeders is, has a really interesting history. He studied at Cambridge and Oxford in the United Kingdom. Then moved to Yale University, was also Howard Hughes Medical Research Institute investigator, and accomplished a lot, published many papers, and sort of jump-starts the whole molecular phase of PKD research. And then actually launched companies, and including a venture capital company that's focusing in the healthcare space. So he has really changed his whole career trajectory and has been just as successful there as well. Dr. Reeders was also instrumental in helping to set up and get up and running Santa Barbara Nutrients in the early days especially, and he's currently serving on the advisory board of Santa Barbara Nutrients. So here is Dr. Reeders. One more thing, this was a really windy day down there by the waterfront. So hopefully you can all hear the voices over the wind noises. So I apologize for that. It might help to turn on the closed captions, the subtitles, to just read along. All right, have fun. Okay. Hello everyone, Thomas Wimes here from Santa Barbara Nutrients. And I'm here in Santa Barbara by the waterfront with my old friend, Stephen Reeders.
Hello.
And Steve is in town for a day here, and I thought we'll just I grabbed the opportunity to have Steve tell us a story of how the PKD1 gene was discovered, and how that all worked, who was involved, and so on. What's the history there? And of course, everybody knows the PKD1 gene is the gene that's mutated in most people with polycystic kidney disease. So that has quite some importance in the whole field. So let's first figure out who is Steve Reeders. You want to tell us a little bit about where you came from originally,
Yep.
and what your training was like, and so on, and what happened next?
Sure, so as you can probably tell from my accent, I'm British, brought up in London, and after my undergraduate degree at Cambridge, I went to learn medicine in Oxford. And I practiced medicine for a handful of years, but one of the formative moments in my medical school career was attending the autopsy of a patient with polycystic kidney disease, and I was asked to write up the story. And I was kind of shocked how little was known about polycystic kidney disease. This was in the early 80s.
Right.
And I couldn't believe, that there was so little research going on in polycystic kidney disease. It was somewhat neglected. And then after practicing medicine, doing some nephrology, I got to the point where I was asked to do some research, and I was in the library at Oxford when Jim Gazella of the Mass General Hospital found the gene for Huntington's chorea, which was the first of the big discoveries. And I thought, wow, I'm a nephrologist. I can do this for polycystic kidneys. I didn't know anything about how labs worked or had no skill, but I thought, how hard can it be, really? I'll just get going on it. And amazingly, my boss, Sir David Weatherall, who was a haematologist, was very positive about it. He said, yeah, you should, why not? I'll support you. Off you go. And that's how I got started.
Ah, that's interesting. Okay, let's turn around real quick just to show everyone where we actually are. Here's the pier of Santa Barbara. So right by the beach. It's a really windy day, but otherwise a beautiful day. And maybe we'll just walk on the pier and
let's do that.
Keep talking. All right, so you're in Oxford. You're a nephrologist. You got this idea. Let's figure out what gene is mutated in polycystic kidney disease. Couldn't be easier than that, right? And so how did you go about it?
Well, the first thing you have to do is to find families. And so I went to the medical records room at the hospital, and in those days there weren't as many rules around privacy. And I got the records out, and I just called people at their houses, and I said, look, you don't know me. I'm trying to sort out the genetics of polycystic kidney disease and maybe find the DNA sequence. And if you'd let me have some of your blood, I'll come to your house, I'll take the blood. And
Really?
I'll
Personally?
personally, and I did all, and I was driving all over the countryside, to the mining districts, and it's very important to collect the whole family. So it's not just enough to have one or two members. You've got to. And I was very fortunate in that a French family, a Protestant French family, moved to the Nottingham area of Britain to escape persecution in the 18th century. And they must have had in that family at that time polycystic kidney disease. And because they were French and they spoke French, they all stuck together. And I found a whole town where there was literally 50 or 60 cases in a small town of polycystic kidney disease. And they would organise parties on a Sunday afternoon, serve tea, and then I would go in the kitchen with my blood taken. And they'd let me all take their blood.
Nice.
And so because of that helpful approach by many, many patients, actually, I was able to collect some very large families. And then I teamed up with a doctor in Holland, in Leiden, Martin Bruning, who was doing the same thing. And we shared the families so we could move quickly. So we banked all the blood and we made the DNA, and then I started to use DNA sequencing sources, which were just becoming available. And I would write to the University of Utah, they had a so-called DNA probe, and they let me have it. And I'd scour the world for these probes to try and identify chromosome, which is PKD1 mutation. And then I found that actually not everyone had the same mutation. There were some, and that in a different location, and that became PKD2. But after quite a short time, I got a little lucky, I suppose, and I managed to map the gene to chromosome 16 for PKD1. And of course, that was a big thing, because there'd only really been two genes mapped like that before.
Wow. Oh, that's amazing. So this was still in Oxford, and I guess you started publishing papers there already?
Yeah, published papers,
and then eventually you moved away from there, right?
Yeah, and so there, I got job offers in various US universities, and what that enabled me to do was raise more money and go much quicker.
Right.
And so obviously once you've mapped a gene, that doesn't tell you an awful lot. You need the gene sequence. That was quite difficult in those days.
Right.
So I moved to Yale, and I was very fortunate at Yale in having a series of fellows who are now, many of them, the leaders in the PKD field.
Like who, for example?
Like Steve Somlow, who's now head of...
He stayed in Yale?
Yale. Greg Gemino, who's NIH.
Right.
Feng Qian, who is in Maryland. Luis Onuchik, who went to Sao Paulo, Brazil. But really astonishingly good people,
Right.
Obviously they're now, you'll know those names because they're leaders in the field. And actually my wife, who is so, who at the time was working on another genetic problem in the kidneys, Alport syndrome, and I managed to. She then became my wife. We were interested in the same world.
Right.
And now she works exclusively on PKD, and has done since 1993.
Right. Good. Wasn't Peter Harris part of
Peter
the
Harris
group as
was
well?
Peter Harris was interesting because he was in Boston,
Right.
moved to my lab in Oxford, and then he's British, and then when I moved to Yale, he didn't want to go, and so he stayed in Oxford and did very well, became
Right.
very successful,
Right.
and of course now he's at the Mayo Clinic and has been for a long time. So you know, whatever happened, I was lucky enough to be at the right place at the right time, and it's very gratifying that so many of the people that worked with me, I can hardly say I trained them, but they were great people, and they've just gone on to great stuff in the PKD field.
Yeah, wonderful. Great. So you moved from Oxford University to Yale University, set up a whole group, started to narrow down the gene, and I guess got it pretty narrowed down.
We got it narrowed down, and then three groups, Greg's group, three groups more or less simultaneously found the sequence.
Right.
Peter Harris had the first piece of it,
Right.
and that was just a bit of the gene, albeit an important bit. And then Greg Jamina's group and my group, I was then being helped by a company called Millennium Pharmaceuticals. We put lots of dollars into it. And so the net result was that we all came up with a sequence. We could confirm the sequence is the right one because we had the same thing.
Right.
And that immediately enabled people to start working using the sequence to find out what the PKD1 gene does. And of course then Steve Somlo, got the PKD2 gene. So, and now there are probably how many, Thomas? But dozens of genes that cause some form of cystic disease, of which PKD1 and PKD2 are the most important.
Right.
But there are many, many others. And someone I introduced to the field, Friedhelm Hildebrandt, who was at Yale with me, and he said, "What do you think I should do for research?" And I said, work on cystic disease of the kidney. And he started off, and now he's the head of genetics at, head of nephrology, at Children's Hospital in Boston. Has been hugely successful.
Right.
So,
Wow, nice. That's the story. Very, very impactful. Real quick, it's getting sunset time over Santa Barbara, so I'm gonna grab a bite to eat soon. But just real quick to finish up the story. So I think clearly just discovering the PKD1 gene and then PKD2 really started off sort of the molecular phase of the research. Now it became clear what those proteins are, the polycystins, and then the race is on to figure out what they do, which we actually still don't really know. So there's still plenty of work to be done. But then you also had a little career change and are now doing something completely different, which is kind of partially how we got connected.
Yep.
And what happened? What did you do?
Well, so I got to Yale, and then I was very interested in finding out how this genetics that I was working on could be used to discover new drugs. And I helped set up a company to do that. And then I moved from that into becoming an investor in healthcare things. And I've been doing that now since 1993, and I bought my own company, MVM, in 1997. And we have raised more than $1 billion to invest in healthcare, and we do every kind of different healthcare, from not just kidney disease. But I do try and, because my wife's in PKD, and over the dinner table in our house, it's a bit odd, we discuss PKD most of the time. And, so I'm very interested in, different approaches to curing PKD, and, trying to lend a hand really to Thomas and others who have, putting a lot of effort into really getting treatments for PKD. and that needs capital, and obviously trying to help on how you get capital into these companies and how you build a company. It's really important that commercial interests move into PKD, because that's what often drives therapeutics forward. So that's what I do. We've had some successes outside the kidney disease area, and I'd be very gratified if we could really make progress in PKD. And I think what Thomas's company, Santa Barbara Nutrix, is doing is one of the most exciting approaches, and that's why I lent a bit of a hand to him to work.
Actually, a little bit more than a bit. So Steve, I would say, was really very, very important getting Santa Barbara Nutrients off the ground. Not only knowing a thing or two about PKD, but also knowing a thing or two about launching biotech companies. So that was just just phenomenal. There are not many other people in the world that have that combination of skills. So I'm really grateful and happy that worked out, and that you helped us get off the ground. And Steve is on the advisory board of Santa Barbara Nutrients, and just invaluable as someone to pick brains on. And it's been a fantastic association. Great. Well, thank you, Steve, for telling us the story of how the PKD1 gene was discovered and who was involved and where it all started and so on. Great. Thanks a lot.
Bye.
Bye.